So now that we've gone over our major enzymes that are going to be involved in DNA replication, we're going to walk through the series of steps that need to occur. So remember that DNA replication is taking place within the nucleus of a cell, and specifically, this all happens during the S phase or synthesis phase of interphase. So the overall goal here is to take the information contained within a double helix of DNA, and we want to end up with 2 double helices of DNA. And because this process is semi-conservative, in these 2 helices, half of the information is going to be our original parental strand, as I'm highlighting here, and the other half is going to be the newly synthesized strand. So, for this whole process to start, we obviously need to be able to get at the information. So we need to unzip this double helix. Helicase is the enzyme that does this. So it comes in a localized region of the DNA molecule, and it separates the 2 strands of DNA. And it does this by simply breaking these hydrogen bonds that are weak and hold those complementary bases together. This localized region where all of this is happening and the molecule is actually unzipped, that is called a replication fork. Okay, so once we have the 2 strands separated, we want to make sure that they don't go back together. And that is the job of these little bubbles called binding proteins. So they glom on to the 2 separated strands of DNA, and they keep them separate so that the other enzymes can come in and perform their jobs. Step 3. An enzyme called primase can now enter this open region of DNA, and it adds a very short segment of RNA that's complementary to the parent strand, and it does this in the 5' to 3' direction. And this short little segment of RNA is called a primer. So that's why we call this enzyme primase. So it adds this short primer, and this is really important because DNA polymerase, which is the enzyme we're going to talk about in a second, that does all of the synthesizing of the new strands of DNA, it can only work if it has an existing stretch of information or nucleotides to build on. So it needs this little primer so it can recognize it and start building. So now that we have this short stretch of RNA in place, our little primer, DNA polymerase recognizes where it needs to come in along this replication fork and begin synthesizing the new strands of DNA. So it uses the information along our parental strand or our template strand, this is this half of information, to recognize which bases it needs to bring in order to form our complementary base pairs. So, if we have an A on our template strand, DNA polymerase is going to make sure a bond forms with a T, our thymine. If it reads a C, a cytosine knows that the bond needs to form with a guanine. So polymerase moves along this parent strand or our DNA template strand forms hydrogen bonds with our complementary bases on this new strand of DNA, and it does all of this in the 5' to 3' direction, moving along the strand. So it brings in complementary bases, forms hydrogen bonds between them, and it also forms the sugar phosphate bonds between the backbone of the molecule. So it does all of these things. And once this whole process is underway, it even will come in and remove our primer and replace it, the information on the primer, because that was RNA, it replaces it with DNA bases. So these nucleotides, the complementary ones that are being paired with our template strand by DNA polymerase, actually are all hanging out within the cells. So those nucleotides come from our diet, so the things that we eat, but they also can be recycled. So once cells die, all of the components that make up DNA can be recycled and used to build new information. So in addition to all of those steps involved in building these new strands of DNA, and remember that this is happening along both parent strands or templates at the same time, DNA polymerase can also proofread. So it moves along pairing these complementary bases, but also checks as it does so to ensure that the bases are pairing the way that they should be. Now, it's not perfect every time. Sometimes there are mistakes that are made, and we'll talk later about why this happens, but essentially in really repetitive areas of DNA where we have base repeats, it's easier for polymerase to make a mistake and not realize that a mistake has been made. So anytime a mistake actually is made, that's called a mutation. So at the very simplest level, a mutation is just simply a new base that appears where a different one was located on our parent strand. Mutations aren't always bad. Mutations can be positive. Most oftentimes they have no effect, and we'll learn more about that later. So this image is just to remind you, I've already mentioned this, but keep in mind that this process of synthesizing our new strand is always done in this 5' to 3' direction. This is happening on both strands. So in one case, because of the way our molecule is unzipped, our DNA polymerase can build the new strand of DNA very easily, smoothly, and continuously. Because we have our helicase, everything's unzipped, and our DNA polymerase in this case is moving towards the left from this 5' to 3' direction, and that's the way that our molecule is being unzipped. On the other side of this DNA helix, the other side of this replication fork, we're still working in the 5' to 3' direction. However, in this case, on this image, our DNA polymerase is moving from left to right. So it can't do this in a continuous stretch because the replication fork isn't open enough for it to do so. So this strand we refer to as a discontinuous synthesis, or a lagging strand, because it simply just takes longer for this whole process to work. So on our lagging strand, what happens is our DNA polymerase can really only make short sections at a time. It has to wait for the helicase to open the replication fork a little further. And these short little segments are called Okazaki fragments, named after the scientists that discovered them. So as you'll notice, we've got lots of primers in this case because DNA polymerase needs that primer to build upon. So our primase has to add a primer each time our helicase unzips. It adds another primer. DNA polymerase can come in, build a little short segment, called that Okazaki fragment. Helicase unzips the molecule more, another primer comes in, so DNA polymerase can build again. So keep in mind we've got this going on, kind of two different directions along our replication fork. So finally, we have enzymes that come in and can remove all those RNA primers. And I've already mentioned that DNA polymerase is one of those enzymes that replaces the RNA with DNA along these primers. But that also needs to happen along all these Okazaki fragments. And then finally, we need the enzyme ligase to come in and seal our sugar phosphate backbone. So we want to make sure that this new strand that is held onto our template strand is firmly sealed. And that's what ligase does. It also can help seal those little Okazaki fragments that were in little pieces. Ligase helps to make sure that it's all strong and held together. The way to remember this is ligate comes from a Latin word meaning to tie together. So ligase essentially ties it all together. So this process of DNA replication, I already mentioned that all of these nucleotides and sugars and phosphates that we need are hanging out within the cell. And this happens very quickly. So it's all happening at about 50 bases per second. That's fast. So essentially, if this were to be happening just at one place in the molecule, it would still take a really long time given how big our genome is. So instead of happening just one stretch, we actually have multiple replication forks that are forming along our DNA helix. And those are called replication bubbles. So here we just have an image of these replication bubbles, so you can see that these, within each single bubble, we can call it a replication fork. But as we look along, here's our fork, but as we kind of think about this fork in a bigger context, knowing that there's many of these happening along a chromosome, along the whole stretch of DNA, we have multiple replication bubbles. And this just helps the process go more quickly. And at the end, once all of these bubbles kind of meet, these little sections between can be replicated, and you end up with our continuous strands of DNA. And I moved this so you can't see both strands, but we end up with 2 double helices. Each of those double helix, because it's semi-conservative, half of it was the parent strand or our template, and the other half is our newly synthesized information. It's the new or the daughter strand. So where we started with 1 double helix, we end up with 2. So this slide just builds upon what I just mentioned on the last one, just to really hammer home this idea that we call DNA replication semi-conservative, because each of these new DNA molecules, so we start with 1, we're going to end up with 2, is really half of an old strand or a parent strand and half new. So here on the left we have our parent molecule of DNA. To make 2 strands of DNA from this 1 double helix of DNA, we of course unzip that and build upon our parent strands, right? So these parent strands are serving as our template. We're using the information on that template to know what new bases need to be brought in, and that's because of these complementary base pairing rules, right? So you can see here, DNA polymerase knows that if there's an A T needs to be bound to it. If there's a C, cytosine, that a guanine needs to be paired with it. Okay? And this is happening in both cases. So we end up with 2 strands instead of 1, but those 2 strands, these daughter molecules, are made up of half parental information and half newly synthesized stuff. And we hope, I've already mentioned, that these are identical. So that's the way that this can happen. And when you're looking at these images, note that which strand here is the template, is complementary, right? They're flipped, they're reversed in the reverse direction because of our anti-parallel nature of the DNA helix. So as I've mentioned, DNA polymerase has the capability to proofread as it's pairing these bases complementary to one another. But it does make mistakes, so it's not perfect every time. But the mistakes are pretty rare. So we've got every 100,000 base pairs, DNA polymerase makes a mistake. And overall in our genome, we have about 3.2 billion base pairs. Remember that once this mistake is made, it can proofread and check for errors. So because of this proofreading, the actual rate that mistakes are actually kept or maintained within our DNA is just 1 in every 10 billion base pairs. And remember that mistake, all it takes is just one switch of a base. It's called a mutation. On the simplest level, that's what a mutation means. And I've already mentioned that mutations aren't always negative things. In society, we tend to think of a mutation as being something that has negative effects. And it can, but most often, this mutation just doesn't have any effect. And sometimes it's positive. But the importance of these mutations, if we didn't have mutations, there wouldn't be change over time. That change over time is simply evolution. And change is good in the sense that under environmental conditions that are changing, if we didn't have these mutations that sometimes confer some benefits with them, the populations wouldn't be able to adapt to that change. These mutations are also what give rise to genetic diversity. And when we have a lot of diversity in our genes and the genome overall, it gives rise to different phenotypes, and that can often be advantageous given changing environments or different environments, different environmental conditions that populations encounter. So in our cells, DNA replication is necessary in order for cells or the nuclei to pass on that genetic information. So remember that replication occurs during interphase, so it's always happening before mitosis or meiosis, the process of passing on all of that information to the next generation of cells. Or in the case of our sperm and eggs, being able to pass on that genetic information to the next generation of offspring or individuals. So an application of this process is called the polymerase chain reaction. So researchers can use DNA replication conducted outside of cells in this biotechnology. So simply called DNA amplification. So amplifying, we want to make lots and lots of copies of genetic information. And the first and best known of these techniques is called the polymerase chain reaction. PCR for short. And PCR uses that enzyme of DNA polymerase to rapidly replicate a specific sequence of DNA in a test tube. So in lab settings. So your textbook goes over a wide range of uses of the polymerase chain reaction. I'm using this in a laboratory setting. And there's also the series of steps that enables PCR to be conducted. So this process can end up, because we're amplifying or making lots and lots of copies of a specific piece of information, it actually yields more than 10 billion copies of our target sequence. And this is really important because in situations where our target DNA sequence is extremely degraded or in just really small quantities, this helps amplify all of that information so that it can be read by computer programs or even observed on a gel, for instance. So a great example of this is in forensic investigations where a lot of samples have really low quantities of DNA. And that could be because the samples, say, at a crime scene have been there for a certain amount of time, or someone cleans up a crime scene and there's just a small amount of evidence available. They can build on what's available, amplify it many times in order to detect and read the information contained within that sequence. So you can match it to another person or match it with someone who is at the crime scene. We also use this technique in wildlife ecology when we're out sampling in the field, for instance, and we want to know what species are, say, using a stream. But you can imagine that the stream is a large area and the animals living within it don't occur that frequently. So we can take samples from the stream, and that's called taking environmental DNA or eDNA, and we can make lots of copies of that information to determine which species are present. So here we have an overview of using PCR, and in this case, we're going to use this technique to identify a virus that is causing a particular respiratory illness. So we first need a target sequence, right? A region of the genome that we know the sequence of. We know it's there. We also need two types of lab-made or lab-constructed information. So we need to have primers, just as we saw before. These primers recognize certain regions or pair complementary with our sequence. And in this case, they're going to match up complementarily with our target sequence. We also need lots and lots of available free nucleotides because we need this part of our building block to make our copies of information. We also need what's called TAC1 or TAC polymerase. And this is a unique type of polymerase that was actually identified in a bacteria that lives in hot springs. Actually, first found them out in the Yellowstone area. And because they inhabit hot springs, they can tolerate really high temperatures. And that is important because in order to actually separate our target sequence, to amplify the sequence, make new copies, we actually apply heat to separate the strands. So this special TAC polymerase is able to tolerate those extremely high temperatures to move on and do its job. So here we have our target sequence, and we're going to do a series of temperature shifts in order to amplify this sequence. So first, we're going to raise the temperature to extremely high level, and what does that do? Well, you could see here, breaks the bonds, breaks the hydrogen bonds, or separates the strands, right? So we don't need our DNA helicase that would do that. The heat simply separates the strands for us. Then temperature turns down in our reaction. Help keep these strands apart. So this is all automated within certain machines. You can specify the temperatures that you want. And once these temperatures are down, the primers that we added in the beginning match up with the single strands based on complementary base pairing rules. How do they do that? Well, remember we have our polymerase. So our TAC1 polymerase is there and is our enzymes that helps connect the primers and then can build on those primers with our free nucleotides, filling everything in. So all of our DNA is added, and this process, where the amplification comes in, is this process is happening over and over and over. So the process is repeated multiple times. So each round of amplification helps build the number of target sequences, right? So the first time, so we're starting with these two, but our next round, so we separate these two and build on them, you end up with 4. And the next round is you're separating each of those 4 and turning them into 2 new ones. Turns into 8. So we're doubling it. We're doubling the amount of sequence that we have each time we go through the round of raising and decreasing the temperatures. So the number ends up, to figure out how many copies we have, we simply take 2, multiplying it by n, and n is just the rounds of amplification, or the times that you go through this increasing and decreasing temperature cycles. So typically we go through 30 cycles of these temperature changes, and that would end up yielding 10 billion copies of the target sequence, right? So you can see how this process is extremely useful in taking small amounts of DNA and creating many identical strands from that information. So another application related to the field of DNA replication and structure, and kind of the next step of when you're thinking about, okay, we've amplified a target sequence, now how do we glean the information that's on that sequence? So that is called sequencing our DNA. So sequencing, or figuring out the series of bases encoded within that stretch of genetic information. So one method of doing this is called Sanger sequencing. And Sanger sequencing was developed in 1977 by a British biochemist named Frederick Sanger. And he just invented a way to determine the base sequence of a small piece of DNA. So this was the first technology to do this, and there are more ways to do it now, but all of them are kind of based on this first technique. Uses the same conceptual basis that Sanger sequencing uses today, and it's still used to sequence individual genes or even check accuracy of certain parts of genomes. So we still use this today. And so how this works It really is determining or deducing a DNA sequence by aligning pieces that differ from each other by the end base. So your textbook shows you an image of this, but essentially you're aligning different segments next to one another that each are just varying by one base at the end. So that helps you kind of build on a series of fragments. So you've got ways to separate our DNA into smaller segments, and this can happen at extreme speeds, and then laying them kind of next to one another to look at differences in their sequences. So also keep in mind that some of these techniques, so using Sanger's method or using other sequencing technologies, and we call this next generation sequencing, or next gen, usually is how we refer to them in science. But essentially, we're using larger DNA molecules and sequencing them at a much faster rate using just different computer algorithms. So I've discussed this kind of chain termination. So we're comparing these different segments, like the bases that are at the end of these fragments. So in this case where we've got these PCR replicates, because we've already talked about amplifying different segments of DNA, and then we can take these fragments, we can cut fragments using certain primers to cut them at regions. And when we cut DNA fragments into different sizes, we can isolate them on a gel. So this is an agar gel, and DNA molecules, you can apply a charge, and the charge, the positive charge, attracts the different sections of DNA along the gel. Sections of DNA, segments of them that are really short, can move faster through the gel, right? They're shorter, so they are going to encounter less resistance as they move through this matrix, and they're attracted to the positive charge. Whereas segments of DNA that are a series of really long string of bases, because they're longer, they're heavier, and they can't move as quickly through the gel. So you run this gel, expose the gel and the DNA fragments to an electric current, let it run for a certain amount of time, and you end up with bands along the gel. And each of these bands are just different segments of DNA that are comprised of different lengths or different numbers of bases depending how they move. And then the other thing we can do is we can tag bases with literal fluorescent dye, right? So we've got this tag that fluoresces in response to when it attaches to an adenine. And in this case, you've got a dye that's fluorescing when the end base is a thymine. And there's our cytosine and guanine. So this flash of, you can apply a flash of light and turns on or makes these fluorescent tags glow in a readout. And the computer algorithms essentially figure out where these tags are, and that's what helps determine this final sequence. So here we just have some DNA sequence data, so you can see what this looks like. This is a first generation DNA sequencing technique, so not a next generation one. But essentially it shows you don't actually get to see those nice fluorescent little labels. That's what the computer algorithm does. What the computer does is then spit out these fancy looking graphs that then shows you the wavelengths that are associated with the different bases. And you can kind of fiddle with the peaks and what those peaks are at with regards to wavelengths to determine the DNA sequence. So as I've already mentioned, we now use these next generation techniques or next gen sequencing. And essentially it's doing all of this at a really, really high rate. So it immobilizes lots of pieces of DNA, and it can read and overlap millions of pieces at once and generate huge amounts of data. So it speeds up our DNA sequencing, and it's built on what we've just covered with that Sanger's invention, but essentially just on a much larger scale. So it can read millions of sequences at once. That's what's called this massively parallel approach. So just reading tons and tons of DNA sequences. And then all of those short little sequences are called reads. So each sequence that we have is called a read. And you end up with lots and lots of copies of these reads and make up this target sequence or genome. And it's done lots and lots of times, and in some cases it's supposed to be done at random locations, or is assumed to be done at random locations across the genome. And because it's done that way, you can get huge amount of coverage of the genome sequences, right? You're just being able to look at a much larger component of the genome than just a single gene, like was initially done for Sanger sequencing. And so that's what has contributed to us learning more about the actual bases contained across our entire genome and genome sequencing of humans and other organisms. The flip side to this is that you end up with huge amounts of data, and for folks that do that now as scientists, you also need huge amount of storage and computer processing or memory availability.